A very warm welcome to our online viewers, and thank you for logging in for today's Science AAAS webinar entitled Addressing the Reproducibility Crisis, Validating Antibodies for Life Science Research. I'm Sean Sanders, Senior Editor for Custom Publishing at Science, and it's my pleasure to be the moderator for today's webinar. Antibodies are invaluable tools in the life sciences. However, their value depends on their specificity and reproducibility, and there are certain challenges that must be overcome in order to properly validate antibody reagents. There are many examples of antibodies with off-target binding leading to erroneous conclusions, some of which will be discussed during this webcast, demonstrating the importance of application-specific validation. 5 alternative strategies for enhancing validation of antibodies will be described during this webinar, based on genetic validation, recombinant expression, independent antibodies, orthogonal validation, and capture mass spectrometry validation. Approaches based on these 5 strategies have been used for streamlined, enhanced validation of more than 10,000 antibodies from more than 30 providers, targeting over 7,000 human proteins as part of the Human Protein Atlas program. The results show a path forward for validating antibodies in an application-specific manner suitable for both providers and users. I'm very pleased to introduce our speakers for today's webinar to you now. They are Dr. Mathias Uhlén and Dr. Cecilia Williams, both from the KTH Royal Institute of Technology in Stockholm, Sweden. Thank you both so much for being on the line with us today. Before we get started, I'd like to share some information for our online viewers. At the top right of your screen, you'll find photographs of today's speakers and a View Bio link, which you can click on to read more details about their background and research. Underneath the slide viewer is the Resources tab, where you can find additional information about products and technologies related to today's discussion, as well as a PDF of the slides. After the speaker presentations, we'll have a short Q&A session, during which we will address some of the questions submitted by our live online viewers. So if you're joining us live, start thinking about some questions now and submit them at any time by clicking the Ask a Question button below the slide window, typing the question into the message box, and then clicking OK. You can also log into your Facebook, Twitter, or LinkedIn accounts during the webinar to post updates or send tweets about the event. Just click the relevant icon at the top left of the screen. For tweets, you can add the hashtag #ScienceWebinar. Finally, thank you to Atlas Antibodies for sponsoring today's webinar. Dr. Uhlén and Dr. Williams are going to be sharing the presenters' podium today, so I'll introduce both of them briefly and then pass it over to Dr. Uhlén. Dr. Uhlén founded the Science for Life Laboratory in Stockholm, Sweden, where he served as director from 2010 to 2015, and he has authored more than 750 publications in bioscience with a focus on the development and use of affinity reagents in biotechnology and biomedicine. He's leading the international effort to create the Human Protein Atlas with the aim of systematically mapping the entire human proteome. Dr. Uhlén is a member of the Royal Swedish Academy of Engineering Sciences, the Royal Swedish Academy of Sciences, the European Molecular Biology Organization, and the National Academy of Engineering, and he is president of the European Federation of Biotechnology. Dr. Williams is a professor of experimental oncology at the KTH Royal Institute of Technology and a visiting professor at the Karolinska Institute in Stockholm, Sweden. Dr. Williams' research seeks to understand how hormones and their receptors influence cancer development, especially the protective effect that estrogen has on colorectal cancer development. She has described the impact of different estrogen receptors on breast, pancreatic, and colorectal cancer, including transcriptomic effects involving long non-coding RNAs and microRNAs. She has also been engaged in the issue of reproducibility in life sciences, raising awareness of methodological problems with DNA sequencing of formalin-fixed samples, microRNA detection technologies, and antibody validation. A very warm welcome to both of you, and now over to Dr. Uhlén to get us going today.
A warm welcome to all of you to this webinar. My name is Mathias Uhlén, and I will be sharing this presentation with Cecilia Williams. So today what we would like to do is to talk about the need for antibody validation, And I will start with that, and then I will hand over to Cecilia talking a case study, which I think is very interesting about antibody validation. And then I will move over and talk about proposal for enhanced antibody validation that we're doing as part of the Human Protein Atlas. But before I start, I just wanted to give you a brief update on what is going on in the Human Protein Atlas Consortium. This is an international effort to map all the human proteins with antibodies. We have now validated more than 50,000 antibodies from more than 50 providers. And the results of this is then presented in an open access knowledge base. A lot of the focus in the last year has been on antibody validation, and that's why we think it's very timely to have this webinar. The atlas consists of 3 different parts: a tissue atlas showing where are the proteins in the different parts of a human body, and we use immunohistochemistry and antibodies for this, and this is headed by Cecilia Williams. And the nice thing about using antibodies and immunohistochemistry is that we can get single-cell resolution, and we can see the context of neighboring cells when we're using the antibodies. And if you want to know more about this, there was an article in Science about 3 years ago that described this tissue atlas. Just to give you some examples of how antibodies then are used in the immunohistochemistry atlas. Here are just some examples of proteins in the human kidney, and you can not only see that the proteins are in the kidney, but also if they are in the tubuli, the glomerulus, and even in the substructures of these part of the kidneys. The second part of the Human Protein Atlas is the Cell Atlas. This gives the subcellular profiling, again using antibodies. This is headed by Emma Lundberg, and it again gives you single cell resolution or even subcellular resolution. And if you want to read more about this, there was an article in Science about 1 year ago called The Subcellular Map of the Human Proteome. I just want to give you 2 examples of this sort of data in the protein atlas, in the subcell atlas. And these are 2 proteins which have cell-to-cell variations. They are cell cycle dependent. And you can then beautifully see the staining of single cells and the variation between different cells. For example, the one to the right, you see a protein which sort of moves from the centromere in one cell out to the cytokinetic bridge in the dividing cell, while it's not expressed in the non-dividing cells. So you get a lot of information using the antibodies. The third part of the protein atlas is a pathology atlas, and this was published in August last year, also in Science. And this is very much focused on transcriptomics and relates to what genes has different outcomes in the patients. But we will not talk about that in this webinar. So the Human Protein Atlas now contains 3 different separate parts: the Tissue Atlas, the Cell Atlas, and the Pathology Atlas. But a lot of the focus right now for us is now to validate the data, validate the antibodies, and in the version 18, which was launched in December 2017, a few months ago, we had a whole new chapter of how to validate antibodies, and this is what I will discuss very shortly. So I will then start by talking a little bit about antibody validation and just give you an introduction to this. And obviously there has been a lot of editorials and research articles talking about the problem with antibodies and the vast amount of articles and publications where antibodies has been used in the wrong way. There is definitely not a shortage of antibodies. if you go to the portal Antibodypedia to look for commercially available antibodies, there is almost 3 million antibodies listed in this portal, and for almost 90% of the human proteins, there is at least 10 antibodies listed. So we have a very good supply of antibodies, and it's kind of interesting to note that only for EGFR we have more than 6,000 commercial antibodies available. But obviously the problem is not then the availability of antibodies, but it is the quality and the validation of these antibodies in order to use it for your research. There has been a lot of literature related to antibody validation, and I'm not going to go through that, but you can sort of see that this is a very active community trying then to clean up this field. I do also want to mention a paper that we published a few years ago in Molecular Cellular Proteomics. In this, we tested commercially available antibodies, about 5,000 from almost more than 30 providers, and we checked if these antibodies could go through our standard validation pipeline. And what you can see here is that for some providers, we have almost 100% of them passed our quality assurance criteria, but actually for a few companies there was actually 0 antibodies that actually passed. And obviously the average was about 30% of the antibodies could actually be validated in our quality assurance pipeline. So one of the reasons for that is that an antibody that works for one application doesn't necessarily work for another application. And it's very important to point out that the sample preparation for different applications such as Western blot or immunohistochemistry and so on are very, very different. For example, Western blot, you use detergents. For immunohistochemistry, you use formalin and very, very high temperatures, over 100 degrees, and so on. And obviously, the proteins are affected by this, the epitopes are affected, and the antibodies are affected. So it's very important then to actually validate your antibody in the context and in the application that you want to use it for. So one could say that there are 3 issues for reliable antibody use. One is the target binding. Does the antibody recognize the intended target? Cross-reactivity. Does the antibody recognize other targets? And reproducibility. Will an antibody function predictably over time? And I just briefly want to go through these 3 different issues. The first one is target binding, and it is actually quite reassuring that almost all antibodies that we have tested actually binds to the target. In the Protein Atlas, we do this by a microarray with a lot of proteins, and we now have actually tested almost more than 50,000 antibodies. And I would say that most antibodies bind to the intended target, so usually this is not the problem. The real problem is cross-reactivity. So the antibody binds to the target, but it also binds to unrelated targets. And if those unrelated targets are much more abundant, they will actually give us very high staining, which are false and could give misleading results. And without going into the details, these are 2 publications that clearly show that almost all antibodies, even the ones that are working very nice, they also have some binding also to other unrelated targets. And this is something that one should be always aware of. It's also interesting, on the right side in the protein science paper, we took a polyclonal antibody that works very nice both in Western blots, immunohistochemistry, immunofluorescence, and then we actually made epitope-specific antibodies from those. And some of the antibodies for some epitopes works fine in all assays, Some doesn't work in any of the assays, and some work in some of the applications, but not in others. So it's very, very much depending on what epitope you are actually recognizing. The third issue is reproducibility, and here the recommendation is that all lots should have a resource identifier, that one should always validate each lot and make them publicly available. For polyclonal antibodies, we feel it's very important that the antibodies are purified with the antigen as an affinity reagent. So every antibody in the lot that you're selling or using are actually binding to the target. But also for polyclonals is that every new batch must be validated, use the same validation criteria. But using this, we are quite sure that this issue is fine. I also want to say a few words about polyclonal versus monoclonal reagents. The big difference between those is, of course, that polyclonal recognize several epitopes and monoclonal antibodies only recognize a single epitope. This, of course, has consequences for reproducibility. It's much easier with monoclonal antibodies. But the disadvantage with monoclonals is that it actually only binds one epitope. So the functionality, in our hands, the functionality across applications is usually better with polyclonals, and also the stability, the functional stability of an antibody across sample variation is higher for the polyclonals. So in our hands, we use polyclonals for research applications and monoclonal antibodies for diagnostics and therapeutics, mainly. And before I hand over to Cecilia, I also just want to make one comment, and that is that it's not very easy to say a good or a bad antibody. There are actually bad antibodies that actually don't bind the target, and obviously they are only bad. But then even a good antibody can be bad if you put them in the wrong context of the wrong application. So one should be a little, I think even the word good and bad should be avoided, since this does not reflect the context and application dependence of the antibody use, as was outlined in an article in Nature a few years ago. So with that, I want to hand over to Cecilia Williams, and she will tell you about a case study, how things can go when you have antibodies that maybe are not recognizing the correct target. So, Cecilia.
Thank you. Hello, everybody. I would like to share my lab's experience with antibody validation in our area of research, which is how estrogen receptors function in breast cancer. And to give you a short background to that research, breast cancer have the second highest mortality rate for women in the Western world. And as you can see here, the mortality rate has been remarkably stable since the 1930s, but in the 1990s, this started to improve quite significantly. And this coincided with that Tamoxifen, a drug, was shown in clinical trials to be highly efficacious and was included in the standard clinical care of breast cancer patients. And Tamoxifen binds to the estrogen receptor and inactivates its activity. The estrogen receptor was the first biomarker used in oncology, and it's a treatment predictive marker. It shows which breast cancers express the estrogen receptor, and those can be treated with Tamoxifen, which significantly lowers recurrence, which is shown to the right. The problem is that many patients with time develop resistance, and a third of tumors don't express the estrogen receptor and cannot be treated, and they have a very poor prognosis. So this treatment, there was hope to improve this treatment if needed. So in 1996, a discovery was made at the lab at the Karolinska Institute, and they discovered that we do not only have one estrogen receptor, we have a second one, which is now called estrogen receptor beta. And this was a paradigm shift for the estrogen signaling, understanding of estrogen signaling. And it has since been an intense research area. for over 20 years. And estrogen receptor beta has been found to be expressed in multiple tissues and have a role in many diseases, and over 5,000 publications have been published on the functions or expression of estrogen receptor beta, and companies have been started and clinical trials have been performed. And it has been concluded that estrogen receptor beta has important roles in cancer. So when it was discovered, it was of course a question if it is expressed in breast cancer, and can it help improve the treatment of inhibiting estrogen receptor alpha or in those ER-negative tumors. So estrogen receptor alpha and beta are located on different chromosomes, and they have a similar DNA binding region. They can regulate similar genes, but they are different in the transactivation function. They can have different roles, and they both bind estrogen but are fairly different in the ligand binding domain, and one can selectively activate or inactivate each receptor with small molecules. So they are suitable for therapeutic targeting with receptor selective agonists. So quite early there was a big interest of whether estrogen receptor beta was expressed in breast cancer. And as you can see in this figure, multiple studies have performed studies on clinical cohorts with different antibodies, and they have all found that a quite large fraction of breast tumors do express estrogen receptor beta. Anything from around 25% to 95% express this receptor. And this has been shown with different antibodies and been replicated. So it seems like a quite solid basis for investigating its role. And of note here is that the two antibodies have been particularly well known and considered good, PPG510 and 14C8, and both of them show a high expression of estrogen receptor beta in breast cancer. So during the past 12 years, my group has been involved in trying to understand what estrogen receptor beta does in breast cancer. Should we activate it or inactivate it to help improve the treatment of breast cancer patients? We have studied cell lines to see what its receptor does. The problem is that we have never found expression of ER-beta mRNA in breast cancer cell lines. So what we have done is that we have reintroduced this receptor and studied its function. And then we have found that you should probably activate this receptor, and that it opposes estrogen ER-alpha-driven breast cancer growth. But we have gotten different results, and we have used different methods of overexpression. So we have been a bit concerned about this, but this has not stopped clinical trials from being initiated. So currently there are several clinical trials ongoing targeting estrogen receptor beta in breast cancer or using estrogen receptor beta as an outcome measure. Because we couldn't find it in cell lines, we decided to check it in breast cancer tumors and also in normal breast with qPCR, and we checked in several and still couldn't find significant levels. So when the Cancer Genome Atlas were out, my PhD student, Philip Johnson at the time, decided to look into the expression levels. And we compared with estrogen receptor alpha, which we know in which breast cancer tumors are ER positive. And the TCGA data show that those tumors had high levels of estrogen receptor alpha mRNA, and that those breast tumors which are ER negative, they have significantly lower levels of estrogen receptor alpha mRNA, which is shown in the panel to the left. However, when we looked in the same tumors for estrogen receptor beta mRNA, we could not find any levels. They were all lower than ER-alpha in ER-negative tumors. So this would indicate that these levels of mRNA should not possibly give rise to proteins. And at the same time, multiple groups have found high levels of estrogen receptor alpha protein with immunohistochemistry. So there can be different reasons for this, but one reason we wanted to look into was the specificity of the antibodies that have been used in those studies. So we decided to do an antibody validation. We also needed a really good antibody for our studies. So we collected 13 antibodies, including those most commonly used in the clinical cohorts, and others that have been used in various labs and commercially available, both monoclonal and polyclonal. And then we designed a validation scheme using very well characterized positive and negative controls. We have multiple cell lines where we know we did not have any estrogen receptor beta expressed, neither on mRNA or any functionality. And we had also reintroduced estrogen receptor beta into those cell lines and expressed it and tested its function, so we knew we had it there. And the first thing we did was that we prepared a tissue microarray with several positive controls and negative controls, as well as tissues that had been reported to be positive or negative. And these were all formalin-fixed material. And then we tested this tissue microarray with all 13 antibodies. And to our surprise, 12 out of the 13 ER-beta antibodies could not differentiate between the positive and the negative controls. We found only one that gave a clear signal in the positive control and a negative signal in the negative control. And in parallel, we did the same thing for estrogen receptor alpha, which has a very good antibody, and that showed the expected pattern in all tissues. What we found was that those two most commonly used antibodies in the clinical cohort, PPG510, stayed positive and negative controls the same, and 14C8 could not differentiate the positive controls in a clear manner. So in the next step, we tried the same antibodies on Western blotting, which is different. It's not necessarily that an antibody will work in both assays. But what we found was that PPZ0506, which was the antibody that worked in immunohistochemistry, on our controls, it worked in the Western blot too. It gave a clear band at the expected length in the positive control, and it did not show up anything in the negative control. And here we had also loaded recombinant ER-beta to give the right size in the Western blot. And our positive controls express the protein with a flag tag and should be a little larger. The 14C8 antibody could weakly stain the recombinant ER-beta, but could not really differentiate the positive from the negative control. That showed the band of the correct size in both of them, which would easily be thought to be estrogen receptor beta. And the PPG510 did not work well at all. It did not recognize the recombinant protein, and it stained something completely different in both positive and negative control. Estrogen receptor alpha antibody worked as expected. So next we did an immunoprecipitation with these antibodies and loaded on a gel, cut out different band size bands, and did mass spectrometry. And for PPZ0506, the antibody that had worked in the other assays, we could detect that it bound to ER-beta in the positive control, and it did not bind anything particular in the negative control, which is shown in the lower panel. For the other two antibodies, we could not detect that they actually bound to estrogen receptor beta. It did not show up in the mass spec. For the 14C8 antibody, we found very reproducibly, though, that it bound another transcription factor, PUU2F1, which is also called OCT1 and is known to have roles in cancer. It bound this protein in every replicate and in both the positive and negative controls. So it might be a good antibody for this protein. The PPG510, which has been the most favored antibody in the ER-beta community, actually bound numerous other nuclear proteins, and would, if it binds this in immunohistochemistry, give a nice nuclear staining, but this would probably not be estrogen receptor beta. For ER-alpha, we found that it bound ER-alpha in the positive control and not really anything else in the negative control, as it should. So at this stage, we concluded that we had one antibody, PPZ0506, which performed well in all assays, and we had two that had been used in the clinical cohort who didn't seem to work very well. And the good antibody has never been used in clinical studies. So we decided to test these three over the Human Protein Atlas, which contains tissues from over 44 normal organs and 20 cancer types, and then compare them and compare with their mRNA levels as determined by RNA-seq. And when we did this, we found that the good antibody, the one that had passed our controls, stained only a handful of tissues, which are shown in the panel to the left. And the other two antibodies also stained most of those tissues, but they also stained almost all other tissues as well, which the PPZ antibody did not stain. And next we compared with mRNA levels per RNA-seq, and then we saw that in those tissues that the PPZ antibody stained, we also found ER-beta mRNA, although this receptor is apparently not highly expressed anywhere, but it's mostly expressed in testes and ovary. It's also known that estrogen receptor beta has an important role in female fertility, at least in mice. And in the tissues that the PPZ antibody did not stain, we also found that we could not detect any significant levels of ER-beta mRNA. So it seems like this antibody had a very good correlation to where we could detect mRNA, whereas the two antibodies gave strong and nice nuclear staining in multiple tissues where we could not correlate to mRNA. And we note that, or to corroborate that the mRNA levels are representative in the Human Protein Atlas, we also compared with the GeneText database, where there are hundreds of individual patient samples for each organ, and it showed exactly the same pattern. So we concluded that we had expression in several tissues which could be detected by the PPZ antibody, and we also concluded that this antibody did not stain in breast, normal breast. We did not detect mRNA, and the other two antibodies did stain. So a few tissues can be concluded with a validated antibody to express estrogen receptor beta, and only one cancer type really had high levels of estrogen receptor beta, and that's granulosa cell tumors, a type of ovarian cancer. We also concluded that we could not find any evidence that breast cancer expressed estrogen receptor beta. In the panel to the right, within the red square, the lower two shows breast cancer, and there was no nuclear staining at all by the PPZ antibody. The other two antibodies showed, just like they did in multiple clinical studies, they showed very nice nuclear staining, but we think that our validation scheme shows that this is other proteins that they bind to. So our conclusion here is that our antibody validation challenges not only our own research of estrogen receptor beta and breast cancer, but also the understanding of estrogen biology, because the antibodies used in the literature were deemed unspecific, and the very specific antibody that we found has not been previously utilized. And we believe that this could be a reason why we find publications stating that estrogen receptor beta agonists does not work, as they don't improve symptoms as expected. And we also think that current clinical trials may want to review the underlying basic mechanism for their intended outcome. And on top of that, we think that there might have been a high cost of using inadequate estrogen receptor beta antibodies, and lots of research into tissues and diseases where actually maybe the receptor is not expressed. And we published this last year, and there are more details in this publication if you are interested. And these are not the first case where with the possibility of antibodies contributing to high cost in research reproducibility. Our take-home message is that we want to stress how critical it is with validation, and it's also very time-consuming. We find that knowing which controls are positive and negative is very important. A lot of the antibody validations performed on estrogen receptor beta antibodies use the breast tissue as positive control, and if it's not there, then the validation will not work well. It's also a great tool to utilize the protein and cell atlases available to corroborate expression pattern with mRNA over multiple tissues. And as Mathias will talk about, there are other standardized guidelines for antibody validation, which are good to follow. So with that, I just want to thank the people in my group that have contributed and all collaborators that have given very deep insights into estrogen receptor beta biology and antibody validation. And with that, I leave back to Mathias.
Thank you very much, Cecilia, and thank you very much for showing a very informative case study, which is, as Cecilia was saying, is not the only one. There's a lot of data coming out that people have used antibodies thinking they are binding specifically to the target, but actually they are binding something else. So this is, of course, a very big problem, as Cecilia was saying, not only for the cost of using these antibodies, but also for the resources in time for the researchers doing this, and then also that leading research coming into the wrong directions. So in order to actually try to make this a little bit more, to provide guidelines for antibody validation, we formed an ad hoc committee 2015, which we called the International Working Group for Antibody Validation. I was chairing this committee, but I had very, very prominent scientists, both from the United States, Canada, Europe, and Asia in this. And then we spent a year very intensively going through different ways that one can do antibody validation. And the idea was to try to provide guidelines that you can use without actually prior knowledge of the target, but could still validate the antibodies in different ways. And what we published in Nature Methods in 2016 then was a proposal based on 5 conceptual pillars for antibody validation, which I will go through in rather detail in the following. We have continued working on this in the Human Protein Atlas Consortium, and we have then used these pillars, conceptual pillars, and actually modified them slightly into 5 different enhanced validation principles. And when we are using one of these pillars, we then, and if they pass that validation, we call the antibody enhanced validated. And I want to go through all of these 5 validation principles, but before I do that, I just want to say a little bit about how useful they are and in what applications. In the Protein Atlas, our 3 most common application is Western blot, immunohistochemistry, and immunocytochemistry or immunofluorescence. And as you can see on this slide, genetic strategy is one of them, is good for 2 of the applications. We have 2 independent antibodies and orthogonal that can be used for all 3 applications. Recombinant expression we're using in 2 of applications, and the migration capture MS only for Western blots. So these different strategies are being used a little bit different depending on what application you're using. But the main point is that each application, you need to validate them specifically for that application. So I just want to go through these 5 pillars a little bit more in detail. The first one is genetic strategies, which is a very simple in its principle. The expression of the target protein is eliminated or significantly reduced by genome editing, such as CRISPR-Cas, or RNA interference like siRNA. And obviously then you have a cell line expressing the protein, and then as you can see in the Western blot here, you knock out the gene, and then if the band disappears, you have a very good enhanced validation that the antibody is recognizing your target. We feel that this is a very good validation strategy. Another strategy, which is in a way the opposite, is then, which is very useful when you have very little expression of a protein, and that is to recombinantly express it in a cell line. and then compare with as shown here in the Western blot before and after you have expressed it recombinantly. And if the staining then appears, you know, or you have very good evidence that the antibody is binding to the target in the background of all the cell lysate from that cell line. A third strategy that we like very much in the Protein Atlas is the orthogonal strategy. And the principle here is that the expression of a target protein is compared with an antibody-independent method across a panel of samples. And here it's very important that that you can find a panel of samples or cell lines or tissues where you have a variable expression of your target. But if you do have that, you can then compare then the independent methods such as MS or transcriptomics with what you're seeing with the antibody. And this is, and then you do the comparison. As you can see here, one example comparing with the proteomics, the MS, and one comparing with RNA transcriptomics. And in both cases, these antibodies are then validated, enhanced validated. The fourth method, which does not require anything extra in samples and preparations, is to use independent antibodies. And here it's important that you have at least two antibodies and with non-overlapping epitopes, and then you use those across a panel of samples. And ideally then, the target protein has a differential expression, so you can then compare the two antibodies, and if they have similar staining, you actually validate both of these antibodies. And then the fifth and last pillar then is capture MS. And the way we have used this is a little bit special. We call it migration capture MS, and can only be used for Western blots. But what we do is that we run an SDS page, we cut out, in our case, 50 different slices from the blot, and then we run these in an MS and actually then detect the peptides corresponding to the target proteins in that slice. And then we correlate the size of the target protein using the Western blot and an antibody with what we see in the MS. And obviously then, if you have the right size or the similar or identical size, you can then have, we call it enhanced validation using capture MS. So these are the 5 pillars that we are now using in the Protein Atlas. And in the December release a few months ago, we then released it with a new antibody validation page, where you can then see which of our antibodies has one of these enhanced validations. And I'll just show you here one gene or protein, ALDH6A1. And what you can see here is that we have 4 antibodies that we have gone through in the HPA. 3 of these antibodies are enhanced validated for all 3 applications, and one, the one to the left, is only validated for IHC, but doesn't seem to work in Western blots and immunofluorescence. So just to show you a more detailed view of the validation, at the antibody information page, you can then see that all of these 4 antibodies are enhanced validated for IHC. So every enhanced validated antibody, we show the primary data which we are basing the quality assurance on. So in summary then, we have then gone through a lot of our antibodies that has been used in HPA. Right now we have more than 10,000 enhanced validated antibodies, and more than 7,000 have been validated for Western blots. As you can see here, that we have quite a lot of antibodies, both done with the genetic methods, the independent antibodies, the orthogonal strategies, and so on. So we are now continuing this, and hope then that we can continue this and have most or all of our antibodies validated in this enhanced manner. This shows you the progress of the Protein Atlas all the way down from the 2015 release, the first version, and the number of genes and antibodies in red and blue for every version. And as you can see, during the last years, we haven't really added very much on the gene size, and this is, of course, because we've already done 90% of all the human genes, and the rest are very difficult. And you can see that we haven't added very many blue antibodies, or antibodies which are shown in blue bars, but we have actually replaced and changed quite a lot in order to get better quality. And then you can also see in green that we have started then to introduce the enhanced validated antibodies, which is growing rapidly as we speak. So in summary then, I just want to say a little bit about the difference between these validation strategies. The ideal case is, of course, if you can validate them with even 2 or even 3 of these validation strategies. But it is important to point out that they have advantages and disadvantages depending somewhat on the target protein and how it's expressed in the tissues that you want to analyze. So the genetic method has a very high specificity, but the throughput is somewhat low, and you have to, of course, set up a validation strategy for each gene, and obviously you need to have knockdown reagents that are working. So it has high sensitivity, specificity, but rather low throughput. The recombinant expression also has a disadvantage that is relatively low throughput, but it is excellent when you have a hard time to find samples which are expressing the target protein. But the specificity is not as high. Since you're overexpressing, you might actually have good staining, while in your own material it starts to stain also other proteins. A very nice part strategy is the independent antibodies. It has a rather large throughput because you don't have to make any special samples. But the specificity varies, and mainly this is how much the target protein is changing in the different samples. The same thing is true for orthogonal, which you can do in a very high throughput mode, since you can actually do transcriptomics and proteomics for a lot of targets, and even share the data between different labs. But the specificity varies depending on if the target protein is differentially expressed. And then finally, the capture-MS method, it has problems when you have proteins which are sort of in the mid size range, because a lot of proteins are sort of in the middle of the gel, and therefore the specificity is not as high when you are analyzing a protein which is maybe 40K or so. But the throughput can be very high, since you only have to do the mass spectrometry experiments once, and you can actually share the data from different cell lines with other labs. So they have advantages and disadvantages, but the idea for us is that to try to validate each antibody at least in one of these validation strategies. So with that, I just want to sort of finish off and saying that the Human Protein Atlas, we are trying to adhere to the antibody validation schemes outlined by this international working group. And the long-term goal for us is to provide spatial protein profiles for all human proteins using only antibodies validated with this enhanced manner. So with that, I just want to say and close by saying that mapping the human proteins with antibodies is our quest. We have now used and validated more than 50,000 antibodies from more than 50 providers. And all the data and also the validation data is then provided at the antibody information page in an open access manner. So just want to thank the funders. We are very grateful to the Knut and Alice Wallenberg Foundation that has funded a lot of this work, but also to some other foundations, the Novo Nordisk, the Ellen Persson, the Heart and Lung Foundation. And we're also very happy to be collaborating with Elixir, which is an infrastructure for sharing data resources in Europe. There is a lot of people that have been involved in this. We have spent actually close to 1,500 person years on the Protein Atlas, and I'm very grateful for all the people that have contributed to make this a very, very good atlas in my view. So with that, this is my last slide. We are situated in the central of Stockholm, close at the Karolinska Institute campus. We are in the Science for Life lab, which is a national infrastructure for molecular science, and we are very devoted then to provide the community with well-validated antibodies. And with that, I want to thank you for listening, and I hope that you have questions that we can discuss. Thank you very much.
Thank you so much, Dr. Uhlén and Dr. Williams, for your presentations. We are now going to move along to questions submitted by our online viewers. So just a quick reminder to those joining us live that you can submit your questions by clicking the Ask a Question button below the slide window, typing the question into the message box, and clicking OK. So we have received a number of questions, and we'll get to as many as we can in the remaining time for the webinar. So the first question I'm going to pose, I think possibly to both of you, but we'll start with you, Dr. Uhlén, is do you think that the system of the 5 pillars will be implemented by researchers? And if not, what other validation methods do you feel are rigorous enough to validate the antibodies that they are using in their research?
Yeah, it's a good question. There has been quite a lot of activities with different networks to try to see if we can establish a format for antibody validation. So far, it has been very hard to get to a firm decision about what should be the guidelines for journals and for funders and for researchers. So what we are doing is that we're trying then to formulate and show that these 5 pillars is a good way forward. But I think it still is important that the individual researchers also try to validate in any way possible before they just trust that an antibody is binding to the target they think about. So the short answer is that so far I don't think that there are firm strategies for making a formal guidelines for antibody validations. We are still in an exploratory phase, I think, and different groups are trying different ways, but we are certainly going for the 5 pillars.
So just a follow-up to that, are there currently any journals or grant-awarding agencies or in fact the FDA that's requiring any particular validation strategies?
So a lot of the journals are requiring that you show the primary data for the validation, but since it's not actually very firm what that is, I'm not sure actually, and I should ask Cecilia here about what is the status in the journals.
Oh, I think they have become more, they're asking for a little bit more now. They want you to provide not only which antibody, but also the lot number. And if you publish Western blot gels, they want you to provide the full gel as supplementary data, but these are not requested by many journals, but it's moving in that direction, I think.
Yeah, and I could absolutely say that in the research community and the networks that I'm working, the idea would be to have firm guidelines for the journals in the future, but this is not a digital science. An antibody that is very good in one application can be very bad in another application. There are all between that there are gray zones, so one has to be a little bit careful to make very formal scores, for example, for an antibody, since that would sort of give the community a feeling that this is an antibody that works in all applications and in all contexts.
Dr. Williams, let me come to you with this question. This viewer says that the Protein Atlas, Human Protein Atlas, has validated many antibodies, but what about the negative results? Are you planning to publish the results on those antibodies that did not work in the applications tested? Is that information in the Human Protein Atlas?
That's a very interesting question. So we have, as I said, already back in 2010, we have actually analyzed 5,000 antibodies from commercial providers, and only 30% worked. What we have said is that we are not actually publishing negative results because it wouldn't be fair to the providers, because it could still be a very good antibody, but in the pipeline that we are using it, maybe it's it's an antibody that works in Western blot, and we use it in immunohistochemistry. So we have decided that we will not publish the negative results, but all the time when we have an antibody that we publish on the Atlas, we also show all the validation data, the primary data, so researchers can go and look and actually be convinced that we're actually doing a good job. And we also score the antibodies with a reliability score. So some are very highly reliable and some are low. And this is because some targets we actually don't know very much about. So we are then showing the community that this is an antibody. We think it's right, but we still would like to have input from the community if it's correct or not.
So are there any commercial antibody companies that are currently using these pillars for their antibody validation? So
the only company that I know about that are using it is the sponsor of this webinar, which is the Atlas Antibodies. But I know that there are efforts now from, I think, all the major antibody providers to actually work with these kind of pillars. And for example, the genetic methods is actually now being pushed quite by some of the big antibody providers. So I think part of these pillars are being used, but others are not.
Fantastic. So the next question I'm going to put to you is when applying for an NIH grant, researchers are asked to address reproducibility. Has your work characterized standards that are accepted as the gold standard for antibodies?
You mean the enhanced validation? No, not yet. And one should remember that the antibody research antibody field is 50 years old, and we proposed these validation pillars only 2 years ago, and we are still seeing if we're still sort of working and trying to show examples how they can be used, but there is no requirements, as far as I know, from any funders to require different types of validations. But I should also say that we had a very exciting meeting in Asilomar about 1.5 years ago, where journals, funders, and researchers came together and discussed ways to validate antibodies. and it was very clear that also the funders were very interested in actually seeing more of the firm guidelines for them.
So, Dr. Williams, in the clinical space, you presented some data that I think some researchers probably find a little frightening. So, how well accepted is the need for validated antibodies in the clinical space? Is this a message that is getting through to researchers?
You mean before starting clinical trials?
Exactly, yes.
The research leading up to that? Yeah, I think there is a big gap there with the realization for antibody validation. I think it's far too common to expect that the antibody works when you buy it. So I'm not sure it's getting through the way it should. That it's necessary to always validate your antibody, regardless of what the literature says.
Fantastic. Well, unfortunately, we're at the end of our time, so we're going to have to end the webinar here. So it just remains for me to thank our speakers very much for being with us today, Dr. Mathias Uhlén and Dr. Cecilia Williams from the KTH Royal Institute of Technology. Please do go to the URL now at the bottom of your slide view to learn more about resources related to today's discussion, and look out for more webinars from Science available at webinar.sciencemag.org. This particular webinar will be made available to view again as an on-demand presentation within about 48 hours from now. We're interested to know what you thought of the webinar. Please do send us an email at the address now up in your slide viewer, webinar@AAAS.org. Again, thank you very much to both of our speakers today and to Atlas Antibodies for their kind sponsorship of this educational seminar. Goodbye.